Well, hello everyone, and welcome to the I guess April now 2022 BioBuilder showcase for the Idea Accelerator. BioBuilder, our slogan is bringing tomorrow's science into today's classrooms, and the Idea Accelerator program is one of our favorite new ways of doing that. And what we do is introduce students to synthetic biology, to bioengineering, and to the design, build, test, learn cycle. And do that by letting them design their own synthetic biology projects around a topic that they're passionate about, and sort of give them the tools and skills that they'll need to design this project over the course of just a few weeks. And without further ado, we'll get right into the student project.
We decided to focus on inserting more beta-carotene into maize because maize is very popular in poor parts of Africa, and we believe bioengineering is going to produce more beta-carotene could provide people with these areas with more nutrients.
Our system design is made to make maize corn more rich in beta-carotene to provide people in Africa with more nutrients. Our input will be genes from daffodils and a bacteria called Erwina. Our output will result in maize corn with more beta-carotene. The next step is just to figure out how to put the genes from the daffodils and Erwina bacteria into the corn embryo.
The questions that we still have are, is there going to be a color change? Or how exactly would we be able to put the corn in the embryo? Like golden rice, is there a possibility that this corn can become a real genetically modified plant? And the main thing that we liked about this was, as a group, figuring out how we were going to make the corn produce more beta-carotene. And the most interesting part about it was the difficulty in trying to figure out how this would even go. We think this was a very interesting thing to research and learn about, and it's definitely something good to have an experience to learn from.
We decided to focus on self-defoliating cotton because we felt as it was the strongest idea that we had, as well as having a better possible outcome. The system we designed should allow the cotton plant to grow normally and to its full potential, but when the cotton bulbs reach its full growth potential, the plant would send a signal to the rest of the plant to stop production and let the cotton start to self-defoliate. The input that we chose was ethylene, where the plant will grow and then stop to drop its leaves naturally, and the output would be the process of self-defoliation itself. The next step in the design of this system is to figure out how exactly we would practically put in the chassis into the organism that we are wanting to create. Some of the questions that we're still having is that we are not 100% sure how to use the chassis or which one would be the best for this project. What our group liked best about this project is definitely the ability to use our own creativity in this field that we are studying. Our group really enjoyed this experience, and we're really happy we were able to do it.
Focused on carbon digesting and fuel producing bacteria. Carbon pollution is a pressing issue, but we still need to use fuels every day. An environmentally friendly fuel is the solution.
The system we designed digests carbon dioxide and transforms it into fuel. The input is detecting and breaking down carbon dioxide, and the output is it being transformed into fuel. The next step to design the system is figuring out exactly how this would work. After reading Chapter 9, we learned that scientists are already working on a similar objective using Eutropha, which is a bacteria that converts carbon dioxide into energy-storing polymers, so this will be a great chassis for our project.
Wonder what exactly the carbon dioxide would transform into. Can it be directly transformed into fuel, or will it have to be something else first?
My favorite thing about this project was the creative freedom. There are so many possibilities when it comes to helping the world through biotechnology. It was cool to see our final idea develop through research and teamwork. We really enjoyed this experience, so thank you for this learning opportunity.
I want to say that they really did truly embed themselves in this, and the 3 weeks, even though we had spring break in between, was 3 intense weeks. They were really thinking, and I appreciate this Idea Accelerator because I really have taught the synthetic biology all year. So this is actually a senior elective, and they have been phenomenal. I am so impressed with my own students because that was truly all their own work. I've just been in the sidelines kind of helping a little bit along the way, but not doing near what they did. So thank you. This has been so worthwhile for my students.